Welcome to the deep dive. We're here again, ready to sift through a pile of information articles, notes, research you sent our way, and, well, really figure out what's key, what's fascinating, and why it matters to you.
Exactly. And today we're looking at the potential science behind a pretty interesting manual technique.
Right, this method used in something called Yugani correction. And what caught my eye is it's not just about pressing down.
No, the really crucial part seems to be the release. Releasing that pressure, but with a specific timing aimed at getting muscles and tissues to, relax. That release phase is what we're digging into.
Mm-hmm. And our goal here, looking at these sources, is to explore the hypothesis they present, the idea that this technique might actually be working by tapping into how our own tiny mechanical sensors, the mechanoreceptors, function.
That's the plan. We'll look at what these receptors are, where we find them, how they signal changes, and then try to piece together how that release part, maybe combined with timing, could theoretically interact with these systems to help relaxation happen.
Okay, great. Let's start with the basics then. The stat pearl source lays some groundwork. So, mechanoreceptors, what are they fundamentally?
You can think of them as the body's built-in sensors for anything mechanical: touch, pressure, stretch. There's sensory receptors pretty much everywhere, and they respond to these physical forces.
Any kind of push, pull, or stretch.
Right. And the way they work, according to stat pearls, is basically translating that external mechanical force into an internal signal the cell understands. Often involves, little gates on the cell opening up mechanically gated ion channels. It's how your nervous system gets physical information.
And they're not just on the surface, like for touch, are they? The sources mention skin, sure, but also near bone, and really importantly for us today, deep inside our muscles and tendons.
Yes, exactly. And that's where we find some really key players for muscle control and this whole idea of relaxation. When we talk about muscles and tendons, we're often talking about specific types of mechanoreceptors called proprioceptors.
Proprioceptors. Okay, those are the ones that help us know where our arms and legs are without having to look, right?
That's a big part of it, yeah. They give us that sense of body position and movement. Now, the Oliver et al. Source highlights two major ones in muscle tissue: muscle spindles, often called MS, and Golgi tendon organs, or GTOs.
Okay, MS and GTOs. So where do they live and what's their main job?
So muscle spindles, they're actually embedded right inside the main body of the muscle. Their primary role, as Oliver et al. Describe it, is sensing changes in the muscle's length and also, critically, the rate of that change. How fast is it stretching?
Got it. Length and speed of stretch
Yeah.
and the GTOs.
The Golgi tendon organs, they hang out at the junction point, the myotendinous junction, right where the muscle fibers connect to the tendon. And they're sensing changes in muscle tension or load, basically how much force the muscle is pulling with.
Okay, so spindles for length,
Yeah.
GTOs for tension. Do they send different kinds of messages or lead to different outcomes in the muscle?
They generally do, yeah. And this is really interesting for the relaxation idea. Oliver et al. Point out a key functional difference in their wiring. Signals coming from the muscle spindles through nerves called group Ia afferents mostly tend to make connections that excite the motor neurons of that same muscle. They sort of encourage contraction.
So a signal from a spindle might tell the muscle, hey, tighten up.
It can definitely contribute to that, yeah, like
Oh,
the stretch reflex. But signals coming from the GTOs via group IB afferents mostly have connections that inhibit the motor neurons of that same muscle.
inhibit, meaning they tell it to ease off.
Potentially, yes.
Mm.
Activating GTOs can lead to a reduction in muscle activity, which could mean relaxation, or at least a modulation of the force being generated.
Inhibition leading to relaxation. Okay, that sounds very relevant to what this Yugani technique is aiming for.
It certainly aligns with the objective. Now, the next piece is how these receptors actually send their signals.
Yeah.
This brings us to a really critical concept about how they fire. Both Oliver et al. And the S41598 source talk about this. They don't all just signal constantly when stimulated.
Right. This is where the phasic versus tonic idea comes into play.
Exactly. Think of it like this. Phasic receptors, sometimes called rapidly adapting, they're like a flashbulb. They respond really strongly right when a stimulus starts, or when it stops, or when it's changing quickly. But if the pressure just stays constant, their signal drops off fast.
Ah, okay.
The S41598 source actually uses this neat analogy of an artificial sensor they studied. It showed its biggest response during the loading phase and during the unloading phase. And it was really sensitive to how fast the indentation happened.
So they're really tuned into change, especially rapid change, like letting go of pressure suddenly.
That seems to be the key characteristic. Tonic receptors, on the other hand, the slowly adapting ones, are more like a dimmer switch turned on. They keep firing pretty steadily as long as the stimulus is there. They signal the presence and intensity of a sustained pressure or stretch.
And the sources mention this is down to complex stuff at the molecular level, different ion channels maybe, but figuring out exactly which receptor subtypes do what and the precise mechanics of those phasic responses, that's still being
Oh,
actively researched.
oh, absolutely. The molecular details are incredibly complex and not fully understood, particularly how biological sensors achieve that rapid adaptation or phasic response.
Hmm.
But the functional difference is the key takeaway for us. Some signal presence, others signal change.
Okay, let me guess if I got this. We have these sensors, mechanoreceptors. In muscles and tendons, we've got muscle spindles tracking length and speed, GTOs tracking tension, and crucially, some of these, the phasic ones, are really good at detecting change, like a sudden release of pressure.
You've got it.
So now, let's connect it back directly to the Yagani correction technique. Apply pressure, then that specially timed release to get relaxation. How does this mechanoreceptor stuff potentially explain that?
Well, here's where we build the hypothesis based on those principles. Could the release part of the technique be so effective because it specifically triggers a strong response from those phasic mechanoreceptors?
So the act of letting go quickly, that's the signal.
That's the idea.
Right.
Imagine you apply pressure, the receptors adapt to some extent, maybe some tonic ones are firing steadily. Then, whoosh, you release the pressure quickly. For a phasic receptor, that sudden unloading is a significant change. It's like flicking a switch off abruptly.
Like that artificial sensor reacting strongly to the unloading.
Exactly like that. Now think about the GTOs again, the ones that sense tension and tend to have inhibitory effects. If that strong phasic off signal triggered by the release somehow strongly activates the GTO pathway,
Or maybe dials down the signal from the muscle spindles.
right, or maybe a combination. But if it preferentially engages that GTO inhibitory circuit, that would provide a direct neural signal telling the muscle's motor neurons to, well, decrease their activity.
Leading to relaxation.
Yeah.
So the theoretical chain might be time to release, strong phasic receptor signal, maybe influences GTO activity, inhibition, muscle relaxation.
That's the core hypothesis we can construct from these sources, yes.
Yes.
It's taking the known behavior of these sensors and their connections and applying it to explain why a specific action, the timed release, might produce a desired outcome like relaxation. It's a plausible mechanism.
Okay, but there's another layer mentioned in our notes. The one's titled Ruskenfeld talks about timing this release with breathing awareness. How does that fit in?
Ah, yes, that adds a fascinating dimension. This introduces the concept the notes call attention synchronization. The hypothesis suggested here is that by consciously coordinating the physical event, that moment of pressure release with your focus, like being aware of your breath at that exact instant,
You're sort of priming the brain.
you can think of it that way. The idea is that it might enhance how those signals from the mechanoreceptors are processed further up the chain. In the brain, specifically the cortex, it's not just a reflex loop. You're bringing conscious attention to bear on the sensory input.
So, linking the physical release signal with focused awareness might make the brain pay more attention to it, integrate it better.
That's the thinking. It could leverage multiple neural pathways, potentially making the resulting feeling of relaxation more profound or more consciously registered. And we know mechanoreceptor signals do go to the higher brain centers. The Stat Pearls article reminds us they project to the somatosensory cortex and can influence perception.
Like their example of feeling breathlessness being affected by receptors in your breathing muscles.
Exactly. You feel the stretch, the effort, the sensory input changes your conscious perception. So applying that logic, consciously attending to the sensation of release in a muscle, timed with your breath, might amplify that sensory signal's impact on your conscious state of relaxation.
It suggests the timing and awareness aren't just incidental, but maybe a way to kind of boost the signal and the brain's response to it.
Potentially, yes. Integrating the physical and the attentional aspects.
This is really building a compelling picture, but just so we're crystal clear for everyone listening, where does this explanation actually stand? Is this established fact?
No, and that's really crucial to underline. Everything we've just outlined, the idea that the Yagani correction release technique works by tapping into phasic mechanoreceptors, possibly influencing GTOs, and being enhanced by attention synchronization.
Yeah.
This is all currently a hypothesis.
Okay. A theoretical framework based on applying what we know from the research.
Precisely. It's grounded in established principles from the sources we looked at how mechanoreceptors function, their different signaling patterns like phasic and tonic, their neural connections, and how attention can modulate sensory processing. But the direct conclusive proof linking this specific technique's effectiveness solely and definitively to these exact mechanisms. That connection is a theoretical model. It's applying existing science to build a plausible explanation.
And the sources themselves indicate there are still unknowns, right?
Absolutely. The precise molecular nuts and bolts of how mechanical force becomes a nerve signal, mapping exactly which subtypes of receptors, IA, AA, PA, etc., contribute what specific information, especially regarding complex responses like relaxation. That's all still very active research, as Oliver et al.
Mhm.
And as 41598 point out, we're using the best current understanding to build this hypothesis.
Right. So to sum up this deep dive, we've journeyed into the world of mechanoreceptors, these amazing little sensors. We focused on the ones in muscle and tendon, muscle spindles for length, GTOs for tension.
And we looked at how some of them are really good at signaling change that phasic response.
Then we built this hypothesis. Maybe the release phase of this specific Yugani technique acts as a strong change signal for these phasic receptors.
Potentially activating inhibitory pathways, like
Mhm.
those involving GTOs, which could then promote muscle relaxation.
And finally, we layered on that idea of timed awareness, suggesting it might enhance the brain's processing of that release signal, making the relaxation potentially deeper or more integrated.
It's a fascinating possible mechanism pieced together from current knowledge.
It really is. And isn't it intriguing to think about how these subtle, precisely timed interactions with our body's sensory network, even just the off signal when pressure is lifted, might hold a key to unlocking states of relaxation? It definitely leaves you contemplating the incredible sophistication hidden within our nervous system and, well, how much more there probably is to discover about how it all works.